Chapter 5, Molecular Basis of Inheritance. In the previous chapter, you have learned the inheritance patterns and the genetic basis of such patterns. At the time of Mendel, the nature of those factors regulating the pattern of inheritance was not clear. Over the next 100 years, the nature of the putative genetic material was investigated, culminating in the realization that DNA, deoxyribonucleic acid, is the genetic material, at least for the majority of organisms. In class 11, you have learned that nucleic acids are polymers of nucleotides. Deoxyribonucleic acid DNA and ribonucleic acid RNA are the two types of nucleic acid found in the living systems. DNA acts as the genetic material in most of the organism. RNA, though it also acts as genetic material in some viruses, mostly functions as a messenger. RNA has additional roles as well. It functions as adapter, structural, and in some cases as a catalytic molecule. In class 11, you have already learned the structure of nucleotides and the way these monomer units are linked to form nucleic acid polymers. In this chapter, we are going to discuss the structure of DNA, its replication, the process of making RNA from DNA, also known as transcription, the genetic code that determines the sequences of amino acid in proteins, the process of protein synthesis, also known as translation, and elementary basis of their regulation. The determination of complete nucleotide sequence of human genome during last decade has set in a new era of genomics. In the last section, the essentials of human genome sequencing and its consequences will also be discussed. Let us begin our discussion by first understanding the structure of the most interesting molecule in the living system, that is the DNA. In subsequent sections, we will understand that why it is the most abundant genetic material and what its relationship is with RNA. 5.1 The DNA. DNA is a long polymer of deoxyribonucleotides. The length of DNA is usually defined as number of nucleotides or a pair of nucleotide referred to as base pairs present in it. This also is the characteristic of an organism. For example, a bacteriophage known as Phi into 174 has 5386 nucleotides. Bacteriophage lambda has 48502 base pairs BP. Escherichia coli has 4.6 into 10 to the power 6 base pair, and haploid content of human DNA is 3.3 into 10 to the power 9 base pair. Let us discuss the structure of such a long polymer. 5.1.1 Structure of polynucleotide chain. Let us recapitulate the chemical structure of a polynucleotide chain, DNA or RNA. A nucleotide has three components: a nitrogenous base, a pentose sugar, ribose in case of RNA and deoxyribose in case for DNA, and a phosphate group. There are two types of nitrogenous bases: purines, which includes adenine and guanine, and pyrimidines, which include cytosine, uracil, and thymine. Cytosine is common for both DNA and RNA, and thymine is present in DNA. Uracil is present in RNA at the place of thymine. A nitrogenous base is linked to the OH of 1' carbon pentose sugar through an N-glycosidic linkage to form a nucleoside, such as adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, and uridine or deoxythymidine. When a phosphate group is linked to OH of 5' carbon of nucleoside through Phosphodiester linkage, a corresponding nucleotide or deoxynucleotide, depending upon the type of sugar present, is formed. Two nucleotides are linked through 3,5 phosphodiester linkage to form a dinucleotide. More nucleotides can be joined in such a manner to form a polynucleotide chain. A polymer thus formed has at one end a free phosphate moiety at 5' end of sugar, which is referred to as 5' end of polynucleotide chain. Similarly, at the other end of polymer, the sugar has a free OH of 3' carbon group, which is referred to as 3' end of the polynucleotide chain. The backbone of polynucleotide chain is formed due to sugar and phosphates. The nitrogenous bases linked to sugar moiety project from the backbone, figure 5.1. In RNA, every nucleotide residue has an additional OH group present at 2' position in the ribose. Also in RNA, the uracil is found at the place of thymine 5-methyl-uracil, another chemical name for thymine. DNA as an acidic substance present in nucleus was first identified by Frederick Mischer in 1869. He named it as nuclein. However, due to technical limitation in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a very long period of time. It was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. One of the hallmarks of their proposition was base pairing between the two strands of polynucleotide chains. However, this proposition was also based on the observation of Irwin Chargaff that for a double-stranded DNA, the ratios between adenine and thymine and guanine and cytosine are constant and equals 1. The base pairing confers a very unique property to the polynucleotide chains. They are said to be complementary to each other, and therefore if the sequence of bases in one strand is known, then the sequence of other strand can be predicted. Also, if each strand from a DNA, let us call it as a parental DNA, acts as a template for synthesis of a new strand, the two double-stranded DNA, let us call them as daughter DNA, thus produced would be identical to the parental DNA molecule. Because of this, the genetic implication of the structure of DNA became very clear. The salient features of the double helix structure of DNA are as follows. Number 1, it is made of two polynucleotide chains where the backbone is constituted by sugar phosphate and the bases project inside. Number 2, the two chains have antiparallel polarity. It means if one chain has the polarity 5 to 3, the other has 3 to 5. Number 3, the bases in two strands are paired through hydrogen bond H bonds forming base pairs BP. Adenine forms two hydrogen bonds with thymine from opposite strand and vice versa. Similarly, guanine is bounded with cytosine with three hydrogen bonds. As a result, always a purine comes opposite to a pyrimidine. This generates approximately uniform distance between the two strands of the helix, figure 5.2. Number 4, the two chains are coiled in a right-handed fashion. The pitch of the helix is 3.4 nanometer. A nanometer is 1 billionth of a meter, that is 10 to the power minus 9 meter. And there are roughly 10 base pair in each turn. Consequently, the distance between the base pair in a helix is approximately 0.34 nanometer. Number 5, the plane of one base pair stacks over the other in double helix. This, in addition to hydrogen bonds, confers stability to the helical structure, figure 5.3. Compare the structure of purine and pyrimidine. Can you find out why the distance between the two polynucleotide chain in DNA remain constant? The proposition of a double helix structure for DNA and its simplicity in explaining the genetic implication became revolutionary. Very soon, Francis Crick proposed the central dogma in molecular biology, which states that the genetic information flows from DNA to RNA to protein. In some viruses, the flow of information is in a reverse direction, that is from RNA to DNA. Can you suggest a simple name to the process? 5.1.2 Packaging of DNA molecule. Taken the distance between the two consecutive base pairs as 0.34 nanometer, which is 0.34 into 10 to the power minus 9 meter. If the length of DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of base pair with distance between the two consecutive base pair, that is 6.6 into 10 to the power 9 base pair into 0.34 into 10 to the power minus 9 meter per base pair, it comes comes out to be approximately 2.2 meters, a length that is far greater than the dimension of a typical nucleus, approximately 10 to the power minus 6 meter. How is such a long polymer packaged in a cell? If the length of E. coli DNA is 1.36 millimeter, can you calculate the number of base pairs in E. coli? In prokaryotes such as E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell. DNA, being negatively charged, is held with some proteins that have positive charges in a region termed as nucleoid. The DNA in nucleoid is organized in large loops held by proteins. In eukaryotes, this organization is much more complex. There is a set of positively charged basic proteins called histones. A protein acquires charge depending upon the abundance of amino acid residues with charge side chains. Histones are rich in the basic amino acid residues lysine and arginine. Both the amino acid residues carry positive charges in their side chains. Histones are organized to form a unit of 8 molecules called histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome, figure 5.4a. A typical nucleosome contains 200 base pair of DNA helix. Nucleosome constitute the repeating unit of a structure in nucleus called chromatin, thread-like stained colored bodies in nucleus. The nucleosomes in chromatin are seen as bits on string structure when viewed under electron microscope EM, figure 5.4b. Theoretically, how many such beads or nucleotides do you imagine are present in a mammalian cell? The beads on string structure in chromatin is packaged to form chromatin fibers that are further coiled and condensed at metaphase stage of cell division to form chromosomes. The packaging of chromatin at a higher level requires additional set of proteins that collectively are referred to as non-histone chromosomal NHC proteins. In a typical nucleus, some region of chromatin are loosely packed and stains light and are referred to as euchromatin. The chromatin that is more densely packed and stains dark are called as heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. 5.2 The search for genetic material. Even though the discovery of nuclein by Miescher and the proposition for principles of inheritance by Mendel were almost at the same time, but that the DNA acts as genetic material took long to be discovered and proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, and numerous other scientists had narrowed the search for the chromosomes located in the nucleus of most cells. But the question of what molecule was actually the genetic material had not been answered. Transforming principle. In 1928, Frederick Griffith, in a series of experiments with Streptococcus pneumoniae, bacterium responsible for pneumonia, witnessed a miraculous transformation in the bacteria. During the course of this experiment, a living organism, which is bacteria, had changed in physical form. When Streptococcus pneumoniae, pneumococcus bacteria are grown on a culture plate, some produce smooth, shiny colonies, capital S, while others produce rough colonies, capital R. This is because the S strain bacteria have a mucus, which is also known as polysaccharide coat, while R strain does not. Mice infected with the S strain virulent die from pneumonia infection, but mice infected with the R strain do not develop pneumonia. Griffith was able to kill bacteria by heating them. He observed that heat killed S strain bacteria injected into mice did not kill them. When he injected a mixture of heat killed S and live R bacteria, the mice died. Moreover, he recovered living S bacteria from the dead mice. He concluded that the R strain bacteria had somehow been transformed by the heat killed S strain bacteria. Some transforming principle transferred from the heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of genetic material. However, the biochemical nature of a genetic material was not defined from his experiments. Prior to the work of Oswald Avery, Colin MacLeod, and Maclean McCarthy, 1933 to 1944, the genetic material was thought to be a protein. They worked to determine the biochemical nature of transforming principle in Griffith's experiment. They purified biochemicals, proteins, DNA, RNA, etc., from the heat-killed S cells to see which ones could transform live R cells into S cells. They discovered that DNA alone from S bacteria caused R bacteria to become transformed. They also discovered that protein digesting enzyme known as proteases and RNA digesting enzymes known as RNases did not affect transformation. So the transforming substance was not a protein or RNA. Digestion with DNAs did inhibit transformation, suggesting that the DNA caused the transformation. They concluded that DNA is the hereditary material, but not all biologists were convinced. Can you think of any difference between DNAs and DNAs? 5.2.1 The genetic material is DNA. The unequivocal proof that DNA is the genetic material came from the experiments of Alfred Hershey and Martha Chase in 1952. They worked with viruses that infect bacteria called bacteriophages. The bacteriophage attaches to the bacteria and its genetic material then enters the bacterial cell. The bacterial cell treats the viral genetic material as if it was its own and subsequently manufactures more virus particles. Hershey and Chase worked to discover whether it was protein or DNA from the virus that entered the bacteria. They grew some viruses on a medium that contained radioactive phosphorus and some other on medium that contained radioactive sulfur. Virus grown in the presence of radioactive phosphorus contained radioactive DNA but not radioactive protein because DNA contains phosphorus but protein does not. Similarly, virus grown on radioactive sulfur contained radioactive protein but not radioactive DNA because DNA does not contain sulfur. Radioactive phages were allowed to attach to E. coli bacteria. Then, as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. Bacteria which was infected with the viruses that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the virus to the bacteria. Bacteria that were infected with a virus that had radioactive proteins were not radioactive. This indicates that proteins did not enter the bacteria from the viruses. DNA is therefore the genetic material that is passed from virus to bacteria, figure 5.5. 5.2.2 Properties of genetic material DNA versus RNA. From the foregoing discussion, it is clear that the debate between protein versus DNA as the genetic material was unequivocally resolved from the Hershey-Chase experiment. It became an established fact that it is DNA that acts as genetic material. However, it subsequently became clear that in some viruses, RNA is the genetic material. For example, tobacco mosaic viruses, QB bacteriophage, etc. Answer to some of the questions such as why DNA is the predominant genetic material whereas RNA performs dynamic functions of a messenger and adapter as to be found from the differences between chemical structures of the two nucleic acid molecules. Can you recall the two chemical differences between DNA and RNA? A molecule that can act as genetic material must fulfill the following criteria. Number one, it should be able to generate its replica, that is replication. Number two, it should be stable chemically and structurally. Number three, it should provide the scope for slow changes, that is mutation, that are required for evolution. It should be able to express itself in the form of Mendelian characters. If one examines each requirement one by one because of a rule of base pairing and complementarity, both the nucleic acids DNA and RNA have the ability to direct their duplications. The other molecules in the living system, such as proteins, fail to fulfill first criteria itself. The genetic material should be stable enough not to change with different stages of life cycle, age, or with change in physiology of the organism. Stability as one of the properties of genetic material was very evident in Griffith's transforming principle itself, that heat which killed the bacteria at least did not destroy some of the properties of genetic material. This now can easily be explained in a light of the DNA, that the two strands being complementary if separated by heating come together. Wow. This now can easily be explained in light of the DNA, that the two strands being complementary if separated by heating come together when appropriate conditions are provided. Further, 2' OH group present at every nucleotide in RNA is a reactive group and makes the RNA labile and easily degradable. RNA is also now known to be catalytic, hence reactive. Therefore, DNA chemically is less reactive and structurally more stable when compared to RNA. Therefore, among the two nucleic acids, the DNA is a better genetic material. In fact, the presence of thymine at the place of uracil also confers additional stability to DNA. Detailed discussion about this requires understanding of the process of repair in DNA, and you will study these processes in higher classes. Both DNA and RNA are able to mutate. In fact, RNA being unstable mutate at a faster rate. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve faster. RNA can directly code for the synthesis of proteins, hence can easily express the characters. DNA, however, is dependent on RNA for synthesis of proteins. Therefore, the protein synthesizing machinery has evolved around RNA. The above discussion indicate that both RNA and DNA can function as genetic material, but DNA being more stable is preferred for storage of genetic information. For the transmission of genetic information, RNA is better.